interactions I could have also in these kind of breaks and evenings. So now I'm going to present you some results we have about some methods to reconstruct past demographic history. And contrary to previous talks, it will be more like some kind of empirical results, more like an empirical population geneticist. And so this is some work that was done in collaboration with several people listed here. And it will be about inference from the site frequency spectrum. So I guess you all know about the coalescent process. We had several talks about this, but just this slide just to show you that actually, if you have different population histories, so this is some kind of constant population, some population that went through some kind of recent expansion, and if you had some recent population contraction, then you would expect to have very different coalescent trees. And of course, if you add onto these some mutations, actually both the number of mutations and the frequencies of these mutations will also vary depending on these different demographic scenarios. So one thing that you can do is to try to summarize the pattern of diversity by just looking at the distribution of the allele frequencies of in a population, and that's what is usually called the site frequency spectrum. So on this axis, you have the frequency of the different mutations. You have the allele frequencies, and here it's the number of sites at which you see this kind of allele frequencies. So in stationary populations, you have this kind of nice exponential decay of these site frequency spectrum. And if you have expansions, you have a lot of sites that have very low frequencies. And if you have bottlenecks, you have a much more even population. So there is a lot of information you can actually get from this site frequency spectrum in single populations. And you can extend this kind of game by considering the joint site frequency spectrum, where actually here in models that involve two populations or more, in that case in some kind of isolation with migration model, where you have some messenger population that's split into two populations, and these two populations still exchange some migrants. In this case, it's a bit asymmetric. Somehow you would expect, depending on the divergence time or the migration rate between these populations, to see different joint frequency spectra. Okay, so on this axis you have allele frequency in one population. On this axis you have the allele frequency in the other population, and the color scheme corresponds to the different number of sites that have these joint frequencies. So there's a lot of information you can use to try to actually, given these joint site frequency spectra, to reconstruct a bit the history of these populations. So, but as was mentioned before, you have a bit this curse of the site frequency spectrum, as was shown by Sam Myers, Pfefferman, and Nick Patterson. Actually, where actually you can have different demographic histories that can lead to exactly the same site frequency spectrum. And in that case, just show different examples, but just took one. So here you have time, and here you have just a population that can vary in size. And if you have this kind of strange history that goes up and down, somehow you would expect to see the same site frequency spectrum as if you had a constant population size. Okay? So it seems, I think the debate is still a bit open, to which extent we can really reliably infer the past history from site frequency spectrum, and there could be some kind of non-identifiability of problems in some cases. Okay. But despite this, actually many people have tried to still use this site frequency spectrum, and relatively successfully in some cases. And one of the most commonly used methods is this method developed by Kunst in collaboration with different people and Carlos Bustamante. So this program dadi, which is using diffusion approximation to get the expected site frequency spectrum in models that involve up to 3 populations that can diverge and shun migrants, have instantaneous admixture, and relatively complex scenarios. And this has been a bit the most commonly used method to make this kind of inference. And actually, I've been trying to develop some method that could be an alternative to this method there because I wanted to try to estimate scenarios that involve a bit more than 3 populations, a bit more complex scenarios, and there was some kind of problems for inferring this demography in these more complex scenarios using dadi. So, but still, the use of the site-free spectrum is interesting for next generation sequencing data because somehow you can have a huge amount of data and the inference is basically does not depend on the amount of data that you have. So you can have 10 megabases, 100 megabases, or full genome somehow. You just have more precision on this site-free spectrum, and so it scales well with the amount of data. So I think it fits well for this kind of GenGS data sets. The nice thing also is that these methods can be used to study very complex scenarios, as I mentioned, with population divergences, migrations, different population sizes, bottlenecks, expansions. Actually, in scenarios that were before this kind of dadi program, were basically reserved to maybe approximate by using computation methods, but now we can do that with this site-free spectrum. And actually, and these estimations are somehow very fast and could be obtained in minutes, hours, or a few days compared to LBC or full likelihood method that would require weeks or months of computations. So this seems all good, but still there are some problems related to the use of this site-free spectrum. And one of the problems is this: we can get some likelihood, but somehow to maximize this likelihood is not always very trivial. So, and this has to do with different problems, and I'll mention a few of them. But sometimes the approximation of the that is used is not optimal, and sometimes we can have some kind of convergence problem to get to the global optimum. And something that also we don't have is any information about linkage disequilibrium. So it just assumes that all the sites are independent, which is not the case. And to obtain relatively good values, usually you need to repeat estimations from different starting points to be sure that you find this kind of global maximum likelihood or composite likelihood, since it is a composite likelihood because you don't take into account this linkage disequilibrium data. So what also needed is, I said it's good to have, it can be applied to large data sets, but actually you need also some large data sets to apply these kind of methods. So you cannot have gene-specific estimations for these kind of methods because you just don't have enough polymorphic sites in a given gene to apply these kind of methods. So usually you are just trying to estimate genome-wide specific or maybe potentially chromosome specific estimates, but it's difficult to have really estimations on precise regions or too short regions. Okay, so that's maybe one problem. Then another very important problem is that I said, okay, it's good to have next-generation sequencing data, but sometimes if you have low coverage, then you tend to underestimate the low-frequency variants, and I showed you somehow that in some cases, these low-frequency variants have precise signatures for some demographic scenarios, so somehow if you miss those low-frequency variants, then you are likely to actually have wrong inferences about the past history of your populations. So, so some care needs to be done to be used to correctly infer this spectrum, and there are different methods, some developed by Rasmus, some by Peter Kitley and Halligan, that try to give some maximum likelihood estimation with the site-free spectrum, and then we can somehow plug these things after that into these inferential programs. So, okay. So, yeah, I will just show you a bit the framework that I've been using to try to reconstruct this expected site-free spectrum. Actually, it's just based on this very nice idea of Rasmus in this paper of 2000, where it actually showed that these entries of the site-free spectrum can be just obtained as some kind of ratios of expected branch lengths of the coalescent tree. So, for instance, suppose that you have a coalescent tree like this, and you would like to estimate what is the expected frequency for mutations that would have an allele frequency of 2. Basically, this formula just says that actually it's just the ratio of the sum of the branch lengths on which a mutation would lead to 2 terminal nodes over the total tree lengths. Okay? So somehow these entries would just be the sum of these red branches over the total tree lengths. And this is what you should get in expectation, but actually you can easily use simulations to try to approximate this probability by just performing lots of simulations and just summing actually all these branch lengths over all simulations and divided by the sum of all the total branch lengths to get some expected entries in your SFS. And of course, the more simulations you use, the more accurate this estimation can be. And that's really nice because this can be applied to any kind of scenario, and this can be, of course, extended to an arbitrary number of populations. So, and then once we have this expected side-frame spectrum, it's relatively easy to come up with some likelihood, and where you just actually have, in this case, some kind of multinomial likelihood that was proposed by Adams and Hudson. Some dadi actually is using a different likelihood, more Poisson-like, but somehow they are somehow a bit equivalent. And in this likelihood, what you have here is this P0, which is the probability that there is no mutation on the tree, and at the power of the number of monomorphic sites in your sequence times the probability that there is some kind of mutation in your tree at the power of the number of polymorphic sites in your sequence, and then you just have this kind of product of these multinomial coefficients. Okay? So that's a very simple likelihood, and you can, in this case, also use this proportion of monomorphic versus polymorphic sites to somehow calibrate all the tree lengths and to get some absolute values for all these demographic events, divergence times, or population sizes. Okay, so, and then what we did was just to implement all this kind of machinery into some kind of program that would be relatively easy to use, which was an extension of some kind of simulation program that I developed a few years ago. And so we are just using coalescent simulations to estimate this side-transit spectrum. And we use, in principle, a large number of simulations per point to estimate it and to get the likelihood. And the problem might be sometimes that when you have very large numbers of parameters, the problem is to actually optimize, to get optimal likelihood. And for this, instead of having some procedure that tries to optimize all parameters at a time, we have some simple version of this, which is called this kind of conditional expectation maximization scenario algorithm that actually is very simple. It's a bit like an EM, but extended to multiple parameters. So what you do, you're just starting with some kind of random parameter values. You fix all of them except one, and then you maximize for this parameter that you are trying to optimize. And then you take the next parameter, fixing all of them, and then you optimize. And then you cycle like this a number of times until you reach some kind of convergence. Okay? So this procedure seems to work relatively well, and it allows us to explore relatively quickly this parameter space. And actually, the parameters in our case, we just need to specify some kind of search range with a minimum and maximum. But actually, this maximum is unbounded, and we can really explore relatively wide range for all the parameters in our model. So what is also kind of handy with this program is that it can handle, in principle, an arbitrary number of populations, while dadi was restricted to 3 populations. And also something I need to mention is that it can handle these things because actually when we have large number of populations, we are using actually some kind of composite likelihood, because we are just looking at the product of composite likelihood computed between pairs of populations. So we are using just joint self-mixed between pairs of populations, and we're just multiplying all these composite likelihoods to get a global composite likelihood. Because just if you have too many populations, then just this site frequency spectrum itself in multiple dimensions is just exploding. You have billions of entries, and just impossible to estimate all the entries of this multi-dimensional site frequency spectrum. So, but of course, this imposes some penalty, but we didn't find any other way to deal with this large number of populations. So just some example to show you a bit that this approach somehow works. So actually we compared the results we obtained with Statistical with some exact results obtained by Chen. So he had a very nice paper in TPB a few years ago. We actually derived the site frequency spectrum based on some kind of coalescent approach, and he solves actually the site frequency spectrum numerically. But it's restricted to cases where you have no migrations. Okay? So for each kind of population demographic model, then you can get a specific expected site frequency spectrum that you can use to compute likelihoods or what you want. Okay? So if you do that just in this simple case, so we have tested with different scenarios, but on this axis here you have this relative error of the site frequency spectrum for each of the entries of the site frequency spectrum, and just that this is just the density of these errors for this scenario when we have very short divergence time or a bit more ancient divergence times. And of course, these errors are lower if you increase the number of simulations from maybe 1,000 when you have large errors, but to 1 million simulations for each estimation, where actually the errors are never larger than 5%. Okay, and we have approximately the same kind of behavior for this different divergence time. So, and what is also interesting is that we don't seem to have any bias in here with this method, because we have this kind of nicely symmetrical distribution around 0. Okay, so in most instances of what we're going to show you, I used actually 100,000 simulations to get the expected site frequency spectrum per parameter conditions, which seems to be a relatively nice compromise between really accuracy and computing time. So just to show that it worked, I will just show you some results I got by comparing this method with dadi. And this is for different scenarios. The first scenario is this kind of bottleneck model, where we have a population that sometime in the past went through some kind of bottleneck and then recovered its size. And we want just to estimate these 4 parameters in this model. The red dots is the true value, so it's just simulations. So what we did was to simulate 100,000 sequences of 100 base pairs, of 200,000 short sequences of 100 base pairs, which is about 20 megabase of data. And we simulated 10 data sets. And for each data set, we used 50 runs of either FastSimCal2 or dadi to try to recover these estimates. Okay, and here I'm showing you this box plot represents the distribution of the point estimates among these 10 runs. Okay, so that's the true value. That's the box plot for FastSimCal2, and in blue you have box plots for dadi. So we see that the two approaches seem to reconverge on the right values, that you seem even to have a bit smaller better distributions for dadi, but actually here I just report 9 cases of the 10 for dadi, because in one case among the 50 runs of dadi, no one could actually seem to converge, and I had just to remove this case. That's something that you have sometimes with dadi. You have this kind of convergence problems and you have really strange results. So that's a different scenario. We have just an IM model with 2 populations actually migrants at different rates, and that diverge some time in the past from an ancestral population. And here I have these 6 parameters, and again, we have very similar behavior compared to dadi, but in that case, dadi only converge in 8 cases out of 10, and the 2 other cases are quite bad. And maybe even more complex scenarios, this time with 3 populations, which is a bit the maximum that dadi can handle at the time. And this model is some kind of pseudo model of human evolution, where you would have maybe some population from which 2 populations have diverged. Could be, some European, Asian population. It could be Africa, and there could have been some kind of ancestral bottleneck. And we would like to estimate all these parameters. And in that case, it's seen that our method becomes a bit more robust than dadi, which has relatively a wider range of estimated values. And even though in that case, again, I only show 8 of the 10 data sets because the 2 others are really awful. So it's in that for more complex scenario, if you have some kind of limited allocated time, so we just try to use 50 runs for each dadi estimations, then some runs will never converge. And but still, among the ones that still seem to converge, you can have really strange values sometimes. So our conclusion for all this seems to be that our method seems to do at least as well as dadi for simple cases, and in more complex cases, actually it seems to be a bit more robust. So what is actually nice with this method is that we can extend it to much more complex scenarios than just those I mentioned before. So in that case, I just show you some simulations we did for some kind of relatively complex model where we have 2 continent island scenarios. So we have one continent that sends migrants to different islands, and we assume that we are sampling genes from each of these islands. And each of these islands receives different amount of migrants from these continents. And this continent actually was created some time in the past from one of the islands of this continent. Okay? And again, we are trying to estimate this migration rate between the continent of each of these islands there. Okay, so it's a case where we have like 12 populations, and we are trying to estimate the amount of migration received by each of these islands and the time of this creation of this continent and the time of the creation of this other continent. Okay, and the red values are the true ones. And again, we did 10 runs, and this is a bit the estimates we get. And I was myself a bit surprised that we can really recover these migration rates extremely well, even though they cover maybe 3 orders of magnitudes. And each time for each of these migration rates, we are having exactly the same search range. So that covers, in this case, like 5 orders of magnitude for these migration rates. So it seems that in this case, we have a method that can handle relatively complex cases that could be actually used in real situations. So if you have any questions anytime, just ask, okay? So
what amount of data are you simulating?
The data in this case?
Yeah, how much data?
So it's 20 megabase. It's assumed to be 20 megabase of data. And we have 20, so 10 would be 10 deployed individuals per population. That would be this kind of range. Yes?
So do you need to worry about when you search the parameters, do you need to worry about the order of the parameters that you optimize?
The order?
When you say you fix operators and try to maximize one of the parameters.
Oh, yes. In principle, not. I mean, the people developing this ECM algorithm have checked this, and apparently the order does not seem to matter. Yeah.
You cycle through them all a bunch of times.
Yes. But I always cycle them in the same order and several times, and I usually use like 50 cycles to get some convergence. Yes?
So I mean, this is incredibly powerful technology, obviously. So the sort of like versions of what we've been doing in our lab is a lot of studying allele frequency correlation patterns, very simple summary statistics, and trying to learn about population relationships from that. And then there's been a series of papers recently that have tried to automate the construction of topologies and demographies from this, so figure out how populations are related. So what you're doing here is you're proposing a particular model and fitting parameters under a model. And there's a natural sort of exciting extension of this, which is to use this as a kind of tool, but to actually embed it in a search space, like TreeMix or MixMapper or admixture graph, that actually searches for topologies and parameterizations that make more sense.
Exactly. So in this case, actually, what we've been doing, maybe I'll show you a few examples, is that we are embedding that into some kind of model choice technology. So we're just using these archaic AIC indices as ways to validate or to prefer one model over the other one. And for the one that would be possible to do these kind of things, but we don't have any kind of automated way to explore all potential demographic scenarios, and that would involve an arbitrary number of populations and those kind of things. But
so people have done something related to this with TreeMix and Excoffier.
Yes, sure.
So,
yeah.
So you know, I don't know how much we believe collectively, you know, those searches, but they're interesting. And one of the things that's sort of scary about this method is it's obviously sort of precise. You know, we can find databases of data that, you know, you're never going to get good fit to any real data, I guess, you know, at some level, right? So with any parameterization. So
no, sure. I mean, this is simulated data, so it just shows that if you have perfect data, then you can recover these things extremely well. And then I guess it's just a question of trying to find a scenario that would maximize the fit with your data, actually. And then I think we can use this archaic, I think that's why we propose to use this archaic or some kind of similar indices. You could have, if you were having some kind of variation version, you could use this VIC or these kind of things to compare models, I think.
Well, just a quick question. Is there another panel where you've estimated all the island and continent population sizes and you're just not showing them, or are they given?
So in that case, we assumed that we knew the continent sizes, and in these cases, in these kind of island content models, the parameters just depends more or less of this NM parameter.
Right, so you can't separate.
Yes, no, we cannot separate. So we just need this NM parameter and not separately N and M.
Okay.
Okay, so we first try to apply this methodology to some kind of complete genomics data. Well, in that case, we had like 4 populations, one from Kenya, Luya, some Bantu-speaking population from Kenya, some Europeans, CCU samples, some Yoruba, and some African Americans. And in that case, we tried actually, the goal was to see, can we just actually estimate the admixture rate of Europeans into this African American population from southwest of the United States. Okay, so this population, this data has very high coverage, more than 50x, so we assume that we had no error on the SFS, and actually, but we extracted some SNPs from this data set because we had this constraint that each SNP should be at least 5 kb away from the next one.
Why does that matter to you? Because you want them to be effectively on them?
Yes, to minimize ND. So
is there some kind of, like, often what we do is we kind of do some kind of empirical LD correction, right? Is that practical and possible here, or not something that one can
Yes, actually, you could perfectly do that, I guess. It also depends how you get your confidence interval. I think it shouldn't affect the point estimates, but of course, the confidence intervals, you will need to take into account the fact that you have a linkage between these SNPs, yes. And I guess you can reproduce that.
Because in the allele frequency correlation literature that's used on SNP array data to learn about population relationships, there's these 2 screens of people doing different types of things. Some people thin and some people don't thin. Then there's more sophisticated methods that use LD, like fine structure, but for people who treat sites as unlinked, people thin and people don't thin, and I've never seen anybody convincingly show that thinning is better than not thinning. So it's just sort of, you know, so I don't know.
No, actually, we use this structure here because somehow we then treated these SNPs out as independent, okay, which they are not. But actually, it should be easy to actually simulate all chromosomes take into account the distance between the SNP that you have. I think that would be possible. Okay, so that's the scenario we considered. So this is a more relatively complex scenario, but it's not actually not that complex. So we assumed in that case.
Actually, Laurent, there was a kind of a subtle thing of ascertainment in the thinning that you do. Did you do a conditional on segregating in more than one population, or?
No, not at all. No. No settlement about allele frequencies in any population, just about the locations where these SNPs are found in a genome. So that's the scenario we consider. So this looks a bit complicated, but it is not that much. So we just assume that we have some kind of European continent, and we have actually one island, which is this CU sample from Northern Europe, that receives some migrants from this continent. Okay? And we assume then that this continent has actually contributed to the initial formation of this African-American sample. But these African-American samples could also have received a contribution from some what we call the Niger-Congo populations that actually has contributed to the formation of this Yoruba sample and this Luya sample. Okay, so we have this two-continent island model. This is just because we don't think that it's directly the Yoruba that contributed to this sample, and it's also not this North American that contributed to this one. Okay, so and of course this European sample has diverged from some kind of ancestral African populations, and of course this Niger-Congo population continent also has diverged from this global African continent. And actually, here I just show you one of the models that we evaluated, because we also introduced a non-sampled or ghost population, like Monty introduced yesterday, that could be some Eastern African population that could have contributed also to some extent to the gene pool of this Luya in Kenya. Okay, so we wanted to estimate all the parameters of this scenario. Okay, so it looks a bit complex, and it's only this probabilistic cannot be handled by dadi. So we got this kind of estimates. I will not really go over all of them, but they seem to somehow make sense. For instance, we have this admixture rate in the African Americans, which has been estimated of 70%, which matches precisely what was estimated from allele frequencies from SNP chips, actually. And also have this nice also input of maybe 17% from some potential East African gene pool into this Luya, Bantu-speaking population from Kenya. And we have quite a few other parameters that could be interesting.
Laurent, can I just ask a question about the interpretation of these things? There's no sort of back arrows in this?
Back arrows. So actually, yes, these arrows are NM parameters.
So
that's some kind of
back
island.
arrow was such that you have what's it called? Balance, right?
Yes.
Yeah.
So some kind of so this island receives this number of genes per generation from this population. Okay. These are dashed because they represent instantaneous admixture event. That's how we model them. So they are dashed. So these are with different types of arrows. But these other arrows are actually just some kind of estimation of NM.
Right. So there's an implicit arrow going the other direction. No, there isn't. So yeah.
In that case, no.
So the population sizes are in fact growing then, right? How do you?
Actually,
what's your actual model then, right? If you're having sort of.
In this case, so it's a common assumption. So we have some kind of population size here. So genes can either coalesce within this population or migrate to this population, but they cannot come back. Okay, that's why we don't have arrows in the other direction. Actually, we just assume that genes, once they reach this population, cannot come back in this population, because we assume it's large enough so that this is neglected actually now in our model.
parent is from another population in the previous generation, but you keep it based.
Yes. But of course, there has been some gene flow between the two, but we think that we also assume that there are many other unsampled islands that also contribute to this continent. And actually, once you are on this continent, then there is a very small probability to come back to this.
Laurent?
Yes.
So this looks great. What are your least well estimated parameters? So what things is it very uncertain about? There are some numbers there, I don't know exactly what they mean, but that are quite uncertain. So like there's one that's from 0.04 to 27, for example. So these
Which one?
parameters.
This one here.
Yeah.
Yes, that's this parameter here. So we have point estimate that is very low, but actually in that case, this NM value could be actually very large. So it could be up to 26 microns received per generation in this CEU population.
And some of your islands are much larger than your continent, like the CEU is larger than the continent.
This?
That estimate is 38,000 for that, right?
Yes.
And 14,000 for the continent it's coming
Yes.
from? So is
Yes,
that
I guess.
unlikely?
Yeah. Yes, it's a bit like that. Yes. Yeah. What can I say?
It's trying.
Yes.
So what is just interesting is that for the ASWs, you get a time of is it 16 generations?
Yeah, this is fixed actually. That this orange parameter are fixed parameters that are used to somehow calibrate all the other parameters. Okay, so we assume that it corresponded to maybe 1800 or something like that, at the start of the slave trade or something like that.
I actually find it impressive that in the population size in the ASWs, you find your confidence interval is reasonably narrow, given that you probably had very little drift in these 16 generations. Right? So you actually think of this population size in the ASWs actually represent the post-admixture population, or is it kind of a mixture, if you want, of the it's an effective parameter that takes into account the divergence between the source African population and the source European populations and so on?
It's difficult. So, I think it would be difficult for me to try to defend any of these parameters with very strong conviction. I'm not even sure that given the complexity of this parameter space, that all parameters really correspond to the exact maximum likelihood.
But it's a good point because, this is recent time, 16 generations. Everybody knows that the human population size is not 10,000 now, so it's much bigger. But the African Americans in the Southwest, this is recent history. We know the census size of these populations has been huge, and it's much bigger than the upper bound. That's what Simon just said.
But my impression is that given this very short time, you don't have much coalescent events during this,
right, so
this
the
time.
population size
So
should be, the confidence interval should be consistent you would expect with hundreds of thousands or something, millions.
So it's up to 40,000, so it's a bit bigger. But I wouldn't have that much confidence in this number. So I think this number is enough, perhaps, to prevent any coalescent within this time frame, perhaps.
I see. But
That's
it's
what
somehow it's confident it's not huge. It's confident it's not huge. The method is confident it's not huge.
So there must be enough coalescence happening to say it's
It
not.
must think that there's enough coalescence in this sample.
Am I misreading that, or is the point estimate outside of the confidence interval?
Yes, there is, in that case, yes. So, because what we are doing, we get these confidence intervals by parametric bootstrap. So what we are doing, we are just taking these point estimates, generating samples according to these point estimates, and then re-estimating each time the parameters. And then using this empirical distribution to get the confidence levels. And in that case, yes, it's a bit just outside.
They make the suggestion that it might be some relatives in the Southwest African Americans. So if you take out samples, maybe the number will change. If you resample from the Southwest African Americans, there might be a few relatives in there, and that might produce a few recent coalescences.
Potentially, yes. No, but no, it's certainly possible. But given this very short time frame, I wouldn't have much trust on this selective side. But anyway, the goal was more to see if we can recover this proportion relatively well, and to get other parameters.
You get the really large estimate, upper bound estimates for migration. Are they just like correlated with the population sizes? So, like when you have invert a lot of migration, you just don't have any recent coalescences, and so the migration is all invisible.
Yes, could be.
Like if you plot the point estimates and then, like, yeah, just see if those two parameters are there.
Yes, no, it's a possibility. Yes, no. I don't have very strong opinions on this. So, how much time do I have?
3 minutes, 4 minutes.
3 minutes. So, okay. So I had also one part on the estimations of the parameters based on different types of data instead of sequence data. Maybe some SNP chip that was developed with Nick and David Reich, but perhaps I will skip that maybe to have some.
It's a wonderful data set, guys.
Yes. But anyway, the take-home message is that it's possible to estimate parameters using this nice SNP chip. We just need to model the ascertainment, which is possible in our case. And maybe it just, and here we apply that actually to the scenario of divergence between some Yoruba and San population. And, but I think, okay, I'm obliged to skip. But anyway, we have relatively good consistency between estimations done on one SNP chip, one SNP panel, either the Yoruba or the San panels. But okay, I will maybe skip this because I just wanted to show you some extremely preliminary results that I think are also interesting to you guys. So, and this is the case. In that case, that could have been done perhaps with dadi, but we are trying to look at the case of archaic admixture in modern humans. Okay, and we are envisioning a scenario that is a bit like this. So we would have, actually, we have samples from this neonatal altai, so one individual inbred, and we have a scenario where we have a Eurasian population that would have gone through some kind of expansion some time ago after going through a bottleneck, and this population would have been admixed from some potentially unsampled archaic population that would have diverged some time ago with this altai neanderthal. Okay, that's our scenario. And then we're trying to estimate the size of this altai population, the size of this unsampled neanderthal, this ancestral neanderthal, the ancestral size of humans and neanderthals, the ancestral human population size, and these other parameters. Okay, and for this, we are using some data set that come from complete genomics again, and we are using two samples, so either the Chinese or the Tuscany sample. Okay, and each time we estimate parameters using either one or the other population. And for this data set, we have used only non-coding DNA and non-CPG sites. We have used this altai neanderthal, and actually in this case, that's something that we are working on. We just use the unfiltered VCF files. So we have been discussing these issues. And we again isolated some regions that were at least 5 kb apart, regions of 100 base pairs. And we had like 272,000 such regions. And we have a bit this amount of data on which we are making our estimations. And here we just consider those sites for which the ancestral state could be deduced from the 1,000 genome data. Okay, so that's a bit the data set that we're going to use. And that's the estimates we get. So, and in red or orange, we have estimations we get if we use the Chinese sample, and in blue if we use the Tuscany sample. Okay, so overall, we have extremely good overlaps between the two types of estimates, and these are 95% confidence intervals also for this estimation. So maybe one question of interest is about the admixture rate. In our case, we find something a bit more than 1%, 1.1, 1.3%, but in that case, we cannot really distinguish between if there's been more or less admixture in one of the two populations. Okay, we don't have enough power. Okay, so that's an interesting result. It fits with some of the previous estimates that you guys had. We have extremely small confidence interval for this admixture rate. I'm a bit worried about this. For me, it's too small, but anyway, that's what we have at this stage with this data set. And another thing interesting that maybe is against what we would have thought is about the time of this admixture. So, and we find extremely recent admixture time. Okay? And with relatively small confidence interval as well, less than 1,000 generations. Okay? So depending on what is your favorite estimation of mutation rate, if you take some assumed mutation of 2 times 10 to the minus 8, this points to estimation 2 times smaller than 25,000 years. If you use a slower one that has been vacated recently, it goes up to 35,000 years.
It doesn't double.
No, it's the other assumed rate is 1.3.
What if you use 1 times 10 to the negative 8? Does it double?
Yes.
So it goes to 50,000 years.
Yeah. But anyway, it points to relatively recent time of admixture.
What do you think a generation is? How long is a generation?
25 years.
So most people think it's like now like 29. I don't know. It's interesting. It's not obviously different from the recombination-based estimate, but it's very interesting if it is different.
Yes. Yes. But anyway, and we still to have an extremely narrow confidence interval. So this seems to be extremely well estimated, and so it really points to some very recent admixture. But so in this case, just some point admixture. So we don't know if there had been continuous admixture for a longer period of time. Maybe this time could represent the last event of admixture. That's the kind of thing that we didn't explore yet. Okay? So we have, of course, we'd like to a bit extend these kind of scenarios, allowing for potential continuous gene flow instead of admixture, also allowing the estimation of also Denisovan contribution, or something that would look like Denisovan, to have the co-estimation of different admixture rate from archaics to modern humans. Okay, that's the next steps, and I think that we have this possibility to this kind of things can be done. Okay, so yes, that's a bit what I wanted to say. Just that now we are working on different things, like some kind of way to use multiple testers with the destinations. We are thinking about implementing some maybe MCMC scheme, a bit along the lines laid out by Beaumont and Garrigan. And maybe the next step that's a bit far-fetched would be to extend that to maybe multi-locus site-frequency spectrum, which seems a bit more complicated, but may not be impossible to do. Okay, and then some other crazy ideas about using this potentially pedigree information to constrain the coalescent process, and maybe try to be able to use those pedigrees to simulate the coalescent process through the pedigrees when we have information on some pedigrees for some populations. Okay, so that's all.
So you use this composite likelihood story. Is there any sort of, theoretical understanding of, how well that will behave for the sorts of models that you're working with?
Yeah. So I think that Jotunheim has somehow shown that this composite likelihood should lead to a solution that the point estimations should be somehow the composite likelihood should be maximized at the same position as the full likelihood, actually. But of course, to explore the space, this is a bit different. You could have maybe multiple peaks where you would have single peaks in the case of full likelihood. So I guess maybe that's where we have problems potentially, in that this composite likelihood surface might have different properties than the likelihood surface, and then it might be more difficult to explore.
And also different precision on your estimates. curvature of the
Yes,
surface.
that's
that
why we're doing this kind of bootstrap approach to. We are not relying on some kind of likelihood ratio or approximation of by-square stuff.
But when you redo your bootstrap estimates, that's still done with your composite likelihood.
Right. Yes.
In your model of African history, you had this ghost population going into one of your African populations. Did you purposefully introduce a ghost population in different parts of the tree, or how did you?
No, but we compare this model without this ghost population. And this model is significantly better than the model without this ghost population. Because there was something a bit strange about this Louis population that didn't fit. I mean, there were some things in the sampling space and the way it looked that we have some bad fit in some parts of this sampling space for this population. And this really improves a lot the fit to the observed data. And for the populations, the fit seemed okay, so we didn't introduce that.
All right, let's take one last question. I think there was one over there, right?
Yeah, I was just trying to get an idea of how robust the parameter estimates, for example, are. For example, like if you have the wrong model for like cryptic migration, how parameter estimates of effective population sizes are, or if you fixed like estimated parameters and then seeing how you estimate the parameters you fixed in the first place, like that 16 generations, how well it estimates that. Any ideas of robustness?
I would have some kind of intuitions on some of the parameters, but I could not guarantee to be sure about the influence of misspecification of models on the estimation of some of the parameters. But this misspecification issue is an important issue, but it is a strongly model-based inferential framework. I think all methods of inference have this kind of problems. Even these PSMC things, we assume still that there is a single population. It tries to infer. It seems a bit magical that it can infer all these kind of demographic changes, but also still it is also strongly model-based. So somehow, if you have a different model, then all these likelihoods in this model are somehow wrong as well, and how this can affect the estimates, that also is important. So I think it's the curse of this model-based inference.
All right, let's thank Laurent again.